End of term. Well, I don't
You're officially on the record. Thanks for coming. It's really nice out, so we really appreciate you being here. I'm delighted to introduce Dr. Toni Pak from over the road, I guess, not just up the road, from Loyola. She's from the medical campus, so we can hear a little bit more about what that looks like. Dr. Pak has been studying happy things. Lots of really interesting ways that estrogens regulate the brain. Broadly speaking, we were just talking about how some of her work was initially focused on adolescence and all the cool parallels between different developmental stages, different life histories. And so mainly what I think you'll hear about today is more of the aging stuff. But for folks that stick around afterwards, you're welcome to talk about booze during puberty. So lots of good conversations there, but really cool work that's bridging physiology with neuroscience, with cellular and molecular approaches, especially microRNAs, which we don't get a lot in coursework, so I'm happy that y'all can be here to hear that. Originally from UC Boulder, right? In Colorado, spent a lot of time out there working your way through the UC system before landing in Loyola. So she's been a Chicago person for a long time, 17 years. So we're really happy to have you here. Thank you for making the trip. And please join me in welcoming Dr. Pak.
very much. It's really nice to be here. And I'm going to echo what Dr. Bell just said, that you all made the choice to be here and be indoors on a day like today is really an honor. It's quite amazing that you took the time to show up, and I appreciate that. So I'm hopefully going to make it worth your while. Well, we'll see. I'm going to talk about one story in particular. We have a lot of different projects in my lab, but I'm going to talk about microRNAs and the aging brain. But when I was invited, I thought, do I get to talk about RNA and hormones? And she said, sure. So I said, okay, I'll come. I'm very excited. These are my two favorite things to be able to talk about. And I don't know how many of you heard about microRNAs? Do you know a lot about them? Some of you heard? Great. That's great that you've heard about them. We'll talk a little bit more in depth about them maybe than you've heard before. But really the main focus of the work that I do at Loyola, and as she said, I'm on the Maywood campus. It's out about 10 miles directly west on 290. It's a nice campus. You should come out and visit sometime. But I actually grew up essentially and was trained in a comparative biology department. And so I was really excited to be able to talk about these other and think about other ways of looking at things. I love thinking about evolution. I was trained in a circadian rhythm lab. I did some of my PhD work on the evolution of hormones in aplesia, which is a sea slug. And I think that it's really fascinating how, all of these things can come together. And mainly what I've done is when I first started my graduate work, I was really interested in the process of puberty. And I was especially interested in, is it really the process of puberty that is making all these changes in the brain, or is it just the process of getting older? And that is still a question that's really hard to get at because it's hard to pull those things apart. And then I started kind of broadening it out across the lifespan. Are hormones working the same way across the lifespan? And this is one of my favorite pictures. I don't know how many of you have seen it, but hopefully you'll be able to see that you can see a young woman's face in the picture, and some of you may see an older woman's face in the picture. But the idea is that, what we've learned in molecular biology especially is really a lot of the signaling pathways and the way we think things work has really been figured out in a cell, usually a single cell type. If we're lucky, a 3-month-old male mouse. And we think that that's the way everything works all the time. And if there's really just two things that I would like you to get out of this talk is that hormones, they act completely differently, not just in different tissue types, but they change the way they act as we age and at different life stages, like puberty or menopause and everything in between. And the second thing is that RNA is really cool. So 2 things to get out if you take nothing else away. So estrogens have a lot of positive effects on pretty much every tissue of the body. And the ones that I'm going to call your attention to especially are they can be neuroprotective. So we know that in terms of cognitive processing, learning, and memory, Estrogens are really important for promoting that cognitive health. And I say estrogens because they're a class of steroid hormone. Usually we just say estrogen, and when we're referring to that, we're referring to 17-beta estradiol, which is the primary one during the reproductive years. But we know that they're all, as a class, neuroprotective. And we also know that they're cardioprotective. They regulate cholesterol production in the liver, and that helps promote cardiovascular health. And this is why women during the reproductive years tend to have lower incidence of cardiovascular disease than men. So it's not surprising that the declining estrogens after menopause are going to cause some negative effects. And these include Alzheimer's disease, which has a sex bias in that we know that 2/3 of all patients with Alzheimer's disease are women. And this is not only an increase in incidence, but also the progression of the disease tends to be much faster in women. And it's thought that maybe it plays a role, these estrogens, that they're now declining. And not just Alzheimer's disease, but any age-related dementia is higher in women than it is in men. And in terms of cardiovascular disease, that becomes about equal after menopause between men and women, but stroke increases a lot in women after menopause. And the symptoms of these diseases are very different in older women compared to older men. And how they're treated is essentially the same. And we have been talking for many years about, should we have different treatment strategies? Should we have different diagnostic criteria for this? But circulating hormones are really dynamic. So they have daily rhythms. Almost all of them have a daily rhythm. They also have monthly rhythms, and they have lifetime rhythms. And so how the tissues and the body respond to these changing levels is really important across all of these time spans. So if we look at circulating levels across the lifetime, we can see that right about the time of puberty, in both males and females, we get an increase in circulating estrogens. In females, we get these monthly rhythms that happen that make pretty dramatic changes throughout the months. But then in menopause, the interesting thing is that we have lower levels, let's see if I can get this to work, actually lower levels in women than we do in men after menopause. And this puts the way the body has to respond to these big swings and changes actually is really important for how the brain functions. So because we knew from decades of animal research, primarily it was animal research, that taught us that estrogens were good, there was a very large-scale clinical trial called the Women's Health Initiative Study for hormone replacement therapy. This had they enrolled over 150,000 women into this trial. And they basically wanted to see, well, if we give hormones, is that going to increase quality of life for women, especially cognitively? And there were two paradigms. Women were given either a conjugated equine estrogen known as Premarin, or they were given that estrogen plus a progesterone known as Prempro. And the results of this study were pretty surprising. What happened was the trial was canceled early. It was canceled because women who were in the hormone arm of this clinical trial started dying of heart disease and stroke at a much faster rate. So this was confusing. So the results were mixed. And what really came out of these studies was, when we took a deeper dive into the data, is that timing seemed to matter. So women who got the hormone replacement early postmenopause, within first 5 years or so, they had beneficial effects across the board. And this included even different reproductive cancers. Women who received it more than 10 years postmenopause had primarily detrimental effects. So this was really interesting because it's the same hormone. How is the same hormone acting beneficially at one age of your life and then completely detrimental at another age? And that's really the question that has been driving a lot of the research in my lab now for many years. So there was some follow-ups to this. In terms of cardiovascular, there was an ELITE study done, and they showed that there was a significantly decreased risk of arteriosclerosis with early treatment. There was another study called the KEEP study, and what they found was that there weren't really any significant improvements that they could find, but no adverse effects with the early treatment. And then in JAMA, in 2017, what it was noticed was that physicians were afraid to prescribe hormones to women, and women were afraid to take the hormones. And so this study came out basically saying that hormone therapy 5 to 7 years post-menopause does not increase the risk of death. It's actually beneficial, and now that is the stance of the Endocrine Society as well. And if we look at cognitively, we see that treatment early post-menopause, so this is a meta-analysis of a lot of studies, it actually decreased the odds of developing dementia when you had treatment early, and it was increased odds of developing dementia if you had it late. So all of this together, well, I forgot to tell you here, the MIRAGE study, there was a 65% decrease in Alzheimer's disease, which is huge. And in the Kaiser study, there was a 26% reduced risk of all-cause dementia. And in this study, the Cache County, 20 to 32% risk of reduced risk of Alzheimer's disease. So all of these together told us that there is something going on with the timing. And the question that we asked then is, what are the molecular mechanisms underlying this? Why would it be beneficial at some times than others? So I want to tell you first a little bit about how we know it works. And again, keep in mind, this was done, these things were figured out a long time ago, probably in one cell type or one small 3-month-old male mouse. But nevertheless, this is the classical dogma. So estrogens, they enter the cell freely, they're steroid hormones, they bind to their receptors, and then they act as transcription factors. So that means they're going to turn on genes or turn off genes. So here's our messenger RNA that comes from our activated gene that is then translated into protein, and it is these proteins that then cause all of the tissue-specific responses in the cell. And the story I'm going to tell you today is another way that estrogen can work, and this is through microRNAs. So microRNAs serve kind of a regulatory function. What they do is they will bind to a messenger RNA that's been transcribed and block its translation into protein. So in this way, it's kind of an indirect way that estrogens can influence what's happening, because the transcription is going along just fine. It's the proteins that aren't being made in the end. And so now I have to introduce you to the RNA world. So also in my department, we don't really have any RNA biologists. So I had to make a cake to explain all of the different kinds of RNA. So this is the cake that I made. And this one here is a messenger RNA. So probably you recognize that this Hershey Kiss is a 5' cap. And then we have ribosomes, we have transfer RNA. And this part right here, this is my microRNA that's binding to the 3' untranslated region of my mRNA and causing it to block translation. I don't know how many of the other RNAs on here you can identify. These Rice Krispies, that's CRISPR. You guys all heard of those. I don't know how many of you have heard of circular RNAs, but these are here. Some of you have heard of those maybe. We have long non-coding RNAs or link RNAs. This is my little links here. So a lot of different RNAs represented on the cake. It actually turned out to be a really good way to teach them. They enjoyed that and then they ate the cake. But basically, microRNAs are small regulatory RNAs. So we have a lot of different kinds of RNAs that most of us don't usually think about. And microRNAs are part of these small regulatory RNAs. So if we look at the world of RNA, we see that over here, we have messenger RNA. This is the only one that we consider coding. And it's the only one we've identified so far that can be translated into a protein. But all of these other ones that we consider non-coding, a lot of them play housekeeping roles. So ribosomal RNA, transfer RNA, and a lot of them are regulatory. And these regulatory short non-coding RNAs are what the microRNAs fall under. They're about 22 nucleotides long, and all of these fall under the category of silencing. So they're going to silence the gene that is going to be transcribed. So they were discovered loosely, I guess, by Victor Ambrose, Gary Rubkin, and David Balcombe. And they were working on C. elegans as a model, or Arabidopsis, which is a plant. And these three discovered these in the 90s, and they really didn't gain any traction at all because it's a worm and it's a plant, and this is some strange thing that worms and plants do. But then it turned out that it became more broad, and oh, humans have these too. Okay, now we're really interested. And if you look at all the publications since then, we have about 190,000 of them. My lab started at Loyola at the end of 2006, and I was interested in hormones and microRNAs. There was almost nothing known. And so we've been working on this since then, and we didn't know anything, and there was no real literature base to turn to. Now there's about 2,000 of these papers have to do with microRNAs and estrogen, and the brain, we've only got about 80 of them. And I would say that probably a good chunk of these actually came from my lab. So there's still a whole lot we don't know about how this works in the brain. So I'm going to tell you about how they're made, because this is important for the data that I'm going to show you in a little bit. So they are transcribed from the genome just like any other gene. It's usually RNA polymerase 2 or 3 that transcribes it, and we get this primary microRNA. This primary microRNA can be quite long, it can be thousands of bases long, but then it is cleaved inside the nucleus by an RNase 3 enzyme called DROSHA into this precursor microRNA. It's really just this long stem loop structure. It's exported out of the nucleus, and another RNase 3 enzyme named dicer will cleave it into what we call the microRNA duplex. So now we have two strands, and one of these strands is now termed the 5P strand. It used to be called the guide strand. And then the other one is called the passenger strand, or the 3P strand. And the guide strand is the one that gets loaded onto a protein called argonauts. It's the mature microRNA, and it's the one that's going to silence and prevent translation of the mRNA. The other one usually just gets degraded really quickly. So when you hear that, if you read papers and it says that something regulates microRNA expression, they're almost always talking about this form. This is the form that really does all of the work in terms of how we think about it. And they're talking about measuring this mature microRNA, about 22 nucleotides long. And we showed, and a lot of other labs now have shown, that estrogen will regulate mature microRNA levels. In fact, they can form what we call a microRNA signature in different cell types. So we know what the expression should be. And in breast cancer cells, this microRNA signature can be predictive of whether something is an estrogen receptor positive or estrogen receptor negative breast cancer cell. And that's pretty interesting, because then you can determine how to best treat that particular tumor. We know that estrogens and the receptors are transcription factors, so it stands to reason that they can also regulate these primary microRNAs, and probably the mature would then result from the regulation up here. And indeed, a number of labs have shown that estrogens will regulate primary microRNA synthesis. But then it was also found that they can regulate these other components of the biogenesis pathway, regulating export out of the nucleus. So it seemed that estrogens can regulate these microRNAs at a couple of different levels. And that became a pretty interesting concept as to, well, if it's regulating these mature ones, exactly how is it doing it and where? And I just want to point out that if a microRNA decreases, so we have less of this one, that then means we should have more of its target. And these microRNAs have been shown to target thousands, hundreds, or maybe thousands of different genes. So a single microRNA can regulate a whole lot of different genes in the cell. So we can assume that if it goes down, then the mRNA target, we should see more of it. And reverse, if microRNA goes up, then the mRNA target should go down. So we have this hypothesis in my lab that estrogen might be regulating mature microRNA levels differently in young versus old, and we used female rats for this, based on this timing hypothesis, based on the idea that, okay, what's one way that the same hormone could act differently at different ages? Well, if it's regulating these microRNAs differently, then perhaps that would explain the tissue-specific effects that we're seeing at different ages. So that's sort of the consequence of how this might work. So we used a pretty simple paradigm. We had female Fisher 344 rat that we get from the NIH aging colony. And we can have young ones and old ones. We ovariectomize them to get rid of all of the circulating endogenous estrogens, and then we give it back 1 week later, in this particular paradigm anyway. And then our endpoints here, we're basically measuring mature microRNA expression. Why this is showing up so light colored, but we used a number of different brain regions to measure this. And at this time, this was almost a decade, more than a decade ago now, these data. But we used a microarray to do the work. And there is something called MirBase, and MirBase is a database that you can go to, and it'll tell you all the microRNAs that we know about. At that time, there were 723 that we knew about. And in the brain, we found that most of them were very lowly expressed. So they were likely not that relevant. So we focused everything on these high expression ones, and we decided to delve into these. And what we found was that there were about 34 in the brain, this particular brain region, that were regulated by estrogen. It didn't seem to matter how old they were. There was 21 that was regulated by age, so the microRNA was different at one age compared to another. But there were 9 where we saw a timing effect, and that is that estrogen regulated these differently at one age compared to another. And those are the ones we decided to focus on. So here's an example of LET7I. This is in the ventral hippocampus, which is an important area for learning and memory. And what we see here is that this is our young female controls and our young female with estradiol and our old female control. Really, we see the same levels in all of those, but it was the older females who got hormone back where we saw a huge increase in the microRNA. And that increase then told us that, well, that means that probably all those target genes are now going to be further repressed if this one is going up. We don't know what those are yet. We looked at all the other ones and we saw kind of a similar pattern where we have this timing effect. So summarizing a whole probably decade of work, we found that estrogen does indeed regulate mature microRNA expression in the brain, and that wasn't known at the time. It's age dependent, so how it regulates is going to be different depending on how old you are. It's also brain region specific, so each brain region seemed to behave differently. Same microRNA, differently regulated in different brain regions. And this was the one that I kept kind of fixating on, is that it was only a few. And that seemed to be kind of strange. How is it that it's just a few of these? And again, we're only measuring this mature one. So it's only 22 nucleotides long. It's not able to bind a receptor to it or anything like that. So how is it working? So some of the questions that came up, one was, what are the targets? And that seemed like a really big question. And so I decided to skip that one, at least for now. We have gone back and figured some of these out, but it was hard at the time. And is timing important? So I told you that if you were given hormones early, it had one effect, and if you were given it late, it had a different effect. Well, we were looking at young versus old, so then we have a paradigm where they're all old, but some get estrogens early and some get them later, and that might make a difference. But this is the one that I'm going to really talk about more, is how. How is it that this one hormone can regulate this tiny subset of microRNAs differently? And so it stood to reason. I said, okay, well, it's estrogen, they work through estrogen receptors. This is really a no-brainer. This is the easiest thing to measure. It's got to be regulating the transcript, and it wasn't. Much to the dismay of my graduate students who thought this would be a really fast experiment. And then we thought, okay, well, maybe it's the precursor. And at that time, it wasn't really clear how to measure that. We figured it out, and we measured drosha, and we measured the precursor. And nothing. It was exactly the same. Okay, well, maybe it's over here. It's regulating export. No. Dicer, no. This is the only thing that changed. None of the processing. So, but our changes in this mature could not be explained by any of it. And that was puzzling to us. And then I thought, okay, well, what else do we know about these things? Maybe it's regulating stability and turnover. What are the kinetics like? And kinetics is something that I think people don't pay enough attention to, especially in molecular biology. All of these things have a half-life. They all have a rhythm, how they're being expressed and when. And we really didn't know how to really go about addressing this question, especially in the brain. How do we test this? These things are small. We couldn't do any sort of in situ hybridization or anything like that, looking in the brain. They're too small. So I had a really talented student at the time, John Kim, and he decided that he was going to tackle this for his PhD thesis. And he focused on a specific microRNA called miR9. And miR9 was really a great candidate because it's neuronal specific. It's the most abundant microRNA we have in the brain. We know that if you knock it out, the brain doesn't develop. So it's critical for neuronal development. But beyond that, it seems to be important for maintaining normal neuronal function throughout life, and especially into aging. And it was one of ours that was differentially regulated by age and estrogen. So I said, all right, figure out how to determine what the stability is of this. And there was nothing known when he started this about the stability of this miR9. So this is the sequence of the 5p strand. That's this one. This is the one that should be the most stable, loaded onto Argonaut. Like, okay, we'll start there. The 3p strand we expect is going to be degraded pretty fast. So this is a good way to kind of look at the differences there. And he developed a microRNA degradation assay that allowed us to test this in vivo. So we have our animal model. These are our old rats. And we also had a cell model. These are rat hypothalamic-derived cell lines. And you can just take the lysate and you can incubate it with the full sequence of this microRNA that has a radioactive phosphorus tag to it. And by incubating these together, the idea is, well, there's something in there, some factor X, we don't know what it is, in the lysate that's going to degrade this thing. And then he can incubate those together and then look at different time courses and then run it out on a gel and image the radioactivity. And so that's what he did. And this is kind of an example of what one of these gels would look like. So here's our radioactive. So what you're looking at here is radioactive label. It's radioactivity you're looking at. It's not like a Western blot. So this is the full length, 22 nucleotide long microRNA. And you can see after 15 minutes, we see a little bit less of it. And we start to see these different smaller products come up. So these are the cleaved products. And we knew that it must be being degraded from this side, because if it was degraded from this side, we'd lose our marker and we wouldn't be able to see anything on our gel. So we know it's coming from this side. So it's being chopped shorter and shorter and shorter. And these are shorter and shorter products down here. And so we could measure kinetically the rate of the degradation based on what kind of environment the microRNA was sitting in. And so here's an example of what happened then when he tried this. So that was a cell-lined example. This is what happened when he tried it in a brain. Within 1 minute, everything was gone. And he was like, okay, this is not going to work. It's degrading really, really fast. And it turned out in the hypothalamus, this is our miR9 5p, the one we thought would be really stable. It was gone like, pretty much instantly. We see the degradation products, those seem to be pretty stable. But this is our curve for miR9 5p. miR9 3p, though, stuck around a long time by comparison. And this is the half-life of it, and this is its kinetics. And we thought, well, that's weird. That shouldn't happen. This is our passenger strand. This is the one that's supposed to go away. But that didn't happen at all. And so this is, again, what we were what we're looking at here is the 5p and the 3p. And one thing that I want to point out is that part of the microRNA, this part right here, second to about 7th or 8th nucleotide, is called the seed. And the seed is the part that allows it to bind to an mRNA target. That's where the hybridization comes from. So these two have very different seed sequences, which means they actually target different genes. So the implication here is that if this one is sticking around longer, that means it has a whole nother set of mRNA targets that we have to figure out what they are. So that was one thing that kind of surprised us about the data. And then he measured all the rest of them that we found were regulated by estrogen and age, and it turned out in the exact same tissue sample, same brain region, they all had a different kinetics. So that told us that it probably isn't the same factor, X, degrading all of them. It's got to be something specific to each of them. And somehow estrogen is regulating them differently by age. So this became a lot more complicated than we thought when we start out. And so this is, again, a lot of years worth of work. He also showed it was different depending on brain region. So I'm just going to show you the quantification. This is the preoptic area of the hypothalamus. This is our miR-9 5P. It's gone right away. 3P sticks around a long time. Hippocampus, it's faster in the hippocampus than anywhere else. So our general observations were that in brain tissue, the kinetics were really fast. So that must mean whatever's degrading them is in very high abundance in these. Each brain region was totally different, so that makes it a lot more challenging to work on. And the miR-9 3P was always more stable than 5P. And that was something that nobody had anticipated, and we weren't quite sure what to make of that. So we went back to kind of what we knew was happening. We knew it was being degraded from this end. And the question then became, why is one so much more stable than the other? And we thought, well, maybe it has something to do with this end. So what Chun did next was he decided to take the 3 nucleotides, the UGA that's on the 5P, and he put it on the 3P. And then he took the AGU that's on the 3P and he put it on the 5P. And the idea was that maybe if he did that, all of a sudden 5P would be really stable and 3P would be really unstable if these matter. And it's actually, he found out that that was indeed true. So you can ignore the isomer. This is an endogenous variant of these. But if we look at, this is our original miR-9 5P. It's degraded really fast and 3P sticks around longer. When the modified version, now we have 5P sticking around longer and 3P sticking around for less time. So that told us there was something about the cis elements of this that was really important. And then we thought, okay, we still don't know what this factor is. Could it be a protein? He first diluted the sample out so that we could see it doesn't actually stay stable ever, and it does if you dilute it out. So the protein or whatever it was in high abundance. He treated with proteinase K, it was totally stable. So now we have a better idea. It's a protein. Definitely a protein that's degrading this. That seemed to make the most obvious sense at the time. And so now he wanted to figure out what that protein was. And to do that, he then took the same sequence and instead of radio labeling it, he put a biotin tag on it. And this turned out to be a really interesting experiment because what you can do is you put that biotin on, it'll biotinylate all the proteins that are interacting with the microRNA. You purify those out and you run it on a mass spec. And we are very lucky at Loyola to have a really nice mass spec facility that's part of our department. We bought actually 4 really nice mass specs. And so this gave us really interesting data. What we found was that this miR-9 5P had 9 unique proteins associated with it, and miR-9 3P had 7 associated with it. And the ones that are in blue are already known RNA binding proteins, not known to bind to microRNAs, but they were known. This one, HNRP-A1, was known to be involved in early microRNA biogenesis, so making of the primary microRNA. We decided to go after this one and we knocked it down. This is now in cells. And what we found was that we got rid of the primary microRNA 9. So that told us that this HNRP-A1 was really important for biogenesis of miR-9. And of course I thought, well, this makes sense. Now we get rid of this, we're not going to have any mature, right? No, we had more mature. I was like, okay, this is interesting. But what that told us was that by knocking out this RNA binding protein, we got rid of this transcript, but that RNA binding protein was likely the reason that we were getting such fast degradation. Because when we knock it out, it will stabilize it. So I want to kind of get back to our original hypothesis because so far I've told you nothing about estrogen. I don't know how many noticed. But we were too busy trying to figure out how these things are degraded in the first place and what's degrading them before we could actually ask. This turned out to be a much more complicated question than we thought. So whenever you think about, oh, we'll just see what the hormone does, well, you have to make sure you know how it works first, and then you can figure out maybe what the hormone does. So we thought, okay, now what's happening with estrogen? The hypothesis was that estrogen was regulating the turnover. And it turned out that that was true. And I'm just showing you, this is just briefly, this is already published work, like a lot of this is, but basically it was regulated by time and estrogen, miR9, 5P and 3P in both cells and in animal models. So what we learned is that microRNA degradation, the kinetics are dependent on the brain region, age, and cell type. And the degradation kinetics were different for every single microRNA we looked at. So I think this is a really good lesson in thinking about how we generalize our science. When you're working with different cell models or animal models or brain regions and you're thinking, this is the way things work, you have to think about, have to consider all of these other factors. What is the age? What is the sex? What is the time of day? All of these things are likely going to play a role in the results you get. We also found that the nucleotides at the 3' end were really important, important determinants for the degradation rate, and we think that has to do with unique proteins that are binding to that site. Now we've done experiments where we've removed 6 of the nucleotides, we've removed 9, we've swapped them all out. We're doing them one by one to try to figure out which one is the most important and how is it changing the proteins. And the one that we've identified most recently is HNRPA1. So it's a critical regulator of miR9 transcription and increases the degradation rate. And this one actually is turning out to be a really interesting one because, as Dr. Bell pointed out, we also study adolescent binge drinking, and this HNRPA1 seems to be an important regulator of some of the molecular problems we see with adolescent binge. So it's an interesting RNA binding protein. So our current projects, we're still working on the structural features, trying to figure out each one. How is it determining the kinetics of this? And what proteins then are going to be required for its stability? And our most recent work, recent funding, is looking at this in the context of Alzheimer's disease, because what we are interested in is this sex bias in Alzheimer's disease. And maybe these microRNAs, we think, could underlie some of these timing effects, and maybe it could give us some clues to what's happening in Alzheimer's disease. So we have a mouse Alzheimer's disease model. We run the kinetics on these brains. We also have human Alzheimer's brains from deceased patients that were generously donated. And the kinetics of the microRNAs in these contexts are actually really, really interesting. In our mouse model, it seems that the microRNA kinetics are changing long before we ever see any evidence of disease in the brains. And then lastly, we're interested in how estrogen might be changing the subcellular localization. So this is some work that was recently published, but also ongoing, and that is, we know that estrogen will actually change where the microRNA is located. In order to interact with its target, it needs to be in the cytoplasm, but we find that estrogen will shuttle it to the nucleus, it'll shuttle it to a stress granule and a processing body and all different other organelles. And in that case, we don't know what it's doing there, but it's not interacting with its target. So that's another interesting kind of side project going on right now. And with that, I want to thank you all for listening to my story about microRNAs, all the people in my lab, and I especially want to shout out Amanda here. Amanda is a very proud graduate of DePaul. She came and did a master's degree at Loyola, and now she works in my lab while she's waiting to go to medical school, and all of our funding agencies. So thank you all very much. Questions from the students?
The other things that I forgot to mention in the introduction is that Dr. Pak has also been involved in the master's program that some folks use to transition into medical school. She's involved in medical school admissions and is the chair of the department, right? So it does a lot of teaching medical students. So anybody's interested in thinking about career development and progression and mentoring, a great resource there as well. And we can also faculty can leave and y'all can just stay in Laker and ask questions without us around as well after.
Yeah, I'm happy to answer any questions that you might have about medical school admissions.
You're going to stay?
For now.
Yes, you can stay, Tim.
Are you interested in another career? Yes.
So I don't just know the fact that you're saying it's age-dependent degradation, species-dependent degradation. And the focus is kind of more proximate that you're working on. Is there anything kind of done comparative-wise, evolutionary-wise, along? Maybe it's beneficial in some ways, not beneficial, anything in that world being done by anyone or not?
In terms of estrogen, in terms of microRNAs or
In terms of the degradation rate of
the degradation rates?
You see stability,
yeah.
that makes sense in these situations. You see faster degradation, that makes sense in these situations.
Yeah. That's a really great question. I wish I had the ability to do more comparative. Right now, the only comparisons we've been able to make is mouse, rat, and human. And in terms of the benefits, that goes into figuring out what all the targets are, and there's just so many of them. We have identified a few of them, and it does seem to be that the degradation rate does definitely impact the rhythm of the circadian rhythm of the gene expression in the cell, which we think so. We think these are really modulatory and kind of fine tuning how the cells are going to respond to different stimuli. I wish I could look at fish and other kinds of organisms because
just the fact that circadian rhythm varies across organisms, and if it
absolutely
tied to that, is that somehow connected?
Yeah, I don't know the answer to that. I think probably yes, but I don't know. I have no evidence to back it up. But I would bet anything on that it does make it important. These have really been shown to just be modulators of how the cell functions by kind of deciding, oh, do we need more of the protein or less of the protein right now in response to whatever stimuli. And the stuff we're doing with stress granules, we're looking at a lot of different kinds of cellular stressors and how that impacts what the microRNAs are doing. And some of them end up in these stress granules, basically blocking the translation of those genes until the environment is more suitable for them to be transcribed or to be translated, not transcribed.
I was just confused. Is the miR9 5P and the miR9 3P, are they from the same duplex?
They are the duplex, yes. So they're transcribed from the same gene. So the primary miR9 makes both miR9 5P and 3P. But then the two of them will target different genes.
Is that like a common microRNA thing that both strands are regulatory?
Not always. There are some where the other strand is just degraded really fast and it doesn't do anything and you never see it. And that was actually the central sort of dogma when they were first described how they were made. It didn't think that the other strand ever did anything. And it was given a little asterisk in the beginning, so it was like mir star. Now some of them, it seems that the 5P strand doesn't do anything at all and it's all of the 3P. So we're having to now kind of reevaluate and re-annotate which ones are actually the ones targeting the mRNA. So this one just happens to be unique in that they both seem to do it, but miR9 5P has gone so fast, we don't know that it has much impact compared to 3P.
It's a very interesting study, and learn even more maybe some other talks too. So my question is that it looks like the finding that 5P get degraded much faster than 3P was done in 18-month-old rats.
That was done, yeah, that was done, well, we did it in 18-month-old rats, but also we did it in young and we did it in cells. So we've done it now in a number of different conditions. The one that I showed you first was just the first one we did. But
so basically it doesn't depend on the age and anything, so it's just
the degradation rate.
Yeah.
The degradation rate for that one seems to depend mostly, well, if we look at the young, that's a good question. So we were looking at, for the age stuff in that one, we were looking at the levels of the mature, and we assumed that part of the reason that they changed was because of the kinetics. But we didn't look in young animals for degradation rate, but we did look in old and older. And it does change by age if we look old and older, because we've given estrogen back for a short period of time and then we've given it much later in life. So it does change by age, but we never went back to those 3-month rats.
That's interesting. And also the estrogen treatment and cause go back to humans findings like early menopause versus older.
Right, that's why we did the older ones, yeah.
And also the like LH level could be different at early menopause versus later menopause. So maybe there's some other systemic hormone interference with those effects, even progesterone levels, we don't know.
Yeah.
Probably very low by that time all over, but
Yeah.
LH definitely at early menopause, definitely the level could be pretty high.
Yeah, and FSH too.
Yeah.
Yeah, those are very high. Yeah, certainly there could be different, that could be serving to modulate it. I don't know. I've never, I think there's one lab that I know of at Kent State, I think, that talks about, that has studied something about LH receptors in the brain, but I don't know. I can never figure out how LH gets in there. But it's a big glycoprotein, but yeah, I don't know how that might be impacting it at that time point. And we have done the progesterone studies though, where we've given just progesterone alone to try to look at these things with progesterone, and it seems to be not as regulated by progesterone. And in the rats, it's hard because the progesterone levels actually stay kind of high. So, and I think it's coming maybe from the adrenals because they're overactimized. So I don't know why they would be high.
How are you measuring the amount of microRNA expression?
So the earlier studies we did, so we've done them by Northern blot and we've also done them by qPCR. The qPCR, I think, is a better way to go. You can get much higher resolution and sensitivity with those assays. And now they've done a really, when we were first starting, it was really hard to figure out how to measure these, but now they have, special kits that my graduate students insist on buying to be able to like enrich and measure those in particular. But that was a lot of how they were found, is that those used to just be kind of thrown out because it was part of the junk that ended up in the supernatant.
So for qPCR, then how are you designing primers for this stuff? Because they're like tiny.
They're
like
tiny,
20.
yeah. So the forward primer is essentially the entire microRNA.
Oh,
And then when you do your cDNA synthesis, you're actually adding on a universal tail that you can do your reverse primer to.
okay.
Yeah, it's it's almost the entire thing. Yeah, but Northern blot is a great way to, you can see all three species, the pre and the mature, all in the same blot. But it's a lot more labor intensive, and my students don't like to do it. So I let them do the PCR instead.
Can you tell me a bit more about how you got introduced to microRNA and kind of combined your interest with puberty and adolescence? How that just kind of culminated into this?
That's a really good question. So I did puberty and brain development during puberty as a graduate student and as a postdoc. And when I was a postdoc, I had gone to a conference and I'd heard some stuff about microRNAs, and I didn't know anything about them. I'd never heard of them before. When I was a student, I learned about mRNA, ribosomal RNA, and tRNA. That's it. That's the only RNA that exists. And that's all I knew about. And there was a talk about these microRNAs, and I became pretty fascinated. And then I started just making it a intentional thing to learn more about it. And I started going to RNA-focused meetings and conferences. And that slide I showed with all the different types of RNA, the words, that was a slide that was shown at one of these conferences as the opening session. And I was like, I have never heard of any of these. I don't know what they do. I thought eRNA, what is that? Electronic RNA? It's supposed to be enhancer RNA. But I didn't know at the time. And so I just thought that it was fascinating. And I wondered if in the brain this played a role and what hormones might do. So I think your science kind of evolves with your curiosity. And I was always very much a puberty person. And then now I kind of I decided I need to know about the whole lifespan. So, but that's a really great question. Thank you.
You talked a little bit about your fascination with comparative biology. I want to piggyback off of Dr. Sparks's question. If you could do another study with another model organism other than rats, humans, and mice, what would you choose and why?
Wow, you guys ask really hard questions. I would probably, well, I've actually wanted for a long time to get C. elegans model going in my lab. One, it's because their life cycles are really fast. We know all the neurons, so that seems like it'd just be a really easy way to get at mechanism. But I think really probably zebrafish would be the other way that I would go, just because there's so many great tools that we can do with the zebrafish model. I think it's a great model. So I think those would be the two, but I think it's also harder, especially zebrafish. And as you get to some of those other organisms, you know, if you're relying on things like antibodies, you know, you're not going to find them. The great thing about RNA work is it's just radioactivity is all you need. You don't need antibodies. So you know, you may be afraid to kind of work with it, but I always thought of it as well. I have to wear this monitor every time I work with it. I know exactly how much I've been exposed to. A lot of these chemicals in the lab, I have no idea how much I've been exposed to. So I'm almost more comfortable with the radioactivity than the thing. But yeah, that's a really good question. I think those two, C. elegans and zebrafish would be the models that I would work on. Although I have to say, one of my graduate school, when I was in grad school, one of my cohort good friends, she worked on kokanee salmon as a model for Alzheimer's disease. And I think that that's also a great model. They essentially are one of the few models besides humans, few animals that actually get the tau tangles, the plaques, just as part of their normal lifespan. They basically migrate back to their home, they spawn, they breed, then they die, and they all die of like Alzheimer-like phenotype. So.
Hi, so you mentioned the question of mRNA targets.
Yes.
You didn't talk about it, which seems very reasonable. Is that something that you have looked at?
Yes.
Or is that something that you're waiting on, or just kind of what's up with that?
We definitely have looked at it. It gets exponentially complicated because, as I said, each microRNA can target hundreds of different genes. It's different in the plants. There's a 1-to-1 hybridization ratio that they pretty much have one target. In animals, it's a lot of different targets. But not only that, each gene can have a bunch of different microRNAs that target it. So one gene that we've played with a lot is BDNF, which is a brain-derived neurotrophic factor. And this protein has 5 different binding sites for microRNA 495. So we've gone through and we've kind of tried to figure out, do you need all 5? If we mutate some of them, are some of them more important than others? Do they act cooperatively with each other? We need 5 mRNA 495s to bind, but then they also bind about 10 other microRNAs. So getting at the targets is an incredibly complex thing to look at, but we have definitely started down that path. It's just taking us a long time to one at a time. And BDNF is the main one that we've looked at right now.
Thank you.
So thanks. Yes.
Hi, so I'm sure you've heard of apolipoprotein B
Yes.
for Alzheimer's disease risk. And
Yes.
I was curious, so that functions a lot in cholesterol metabolism. Cholesterol and estrogen are structurally similar, correct?
Well, estrogens are derived from cholesterol. So
Right.
yeah, all steroid hormones are.
So I'm wondering, do you think that there's a possibility maybe that there's some microRNA effect that is like a genotypic effect, maybe affecting individuals that possess that E4 allele versus, like the E3, E2? Just trying to maybe mesh those things together and just wondering if,
That's a really interesting question. So for that risk, it's typically the familial form of Alzheimer's disease that is manifested, right? And those individuals usually develop Alzheimer's disease much younger in life. then, and I don't know that that one actually has much of a sex bias. The form that we know for sure it does is more the spontaneous form, and not related to the ApoE mutation or the allele that's different. But that's, I don't actually know what microRNAs might target that allele and whether or not they are regulated by estrogens. But that's a really, that I'm gonna go and check that right after I leave here. Because
Yeah, I
that's
just
a
know,
really
like, could
important.
microRNAs have like, you know, allele specific effects? Like,
Yeah,
yeah, they could, right?
I mean, yeah, I mean, they're targeting an mRNA, right? So any mRNA that's gonna be transcribed, it could be targeted by the microRNA, so it certainly could. I just, we've pretty much just focused on the spontaneous form of Alzheimer's because that's the predominant one. So I've never really looked into it, but I think that's a really excellent question, so. But yeah, I will look into it, and then I'll let Dr. Bell know what I find. Or maybe you could look into it and let me know, that would be even better. There's always more questions than answers when you do research.
I'm gonna take host privilege here and ask the last question, and then we'll let y'all continue to chat about it.
I'm gonna ask the last question then.
Yeah.
After you.
Yes, chair privilege.
Well, now it's a fun question.
Okay. I'm thinking about what you said about microRNAs being really quickly responsive, really dynamic and response to changing environments. Do you think anything about the dynamism that you're seeing in this model is unique to brain, or is this just a cool model in which to study something? Do you think the concepts and the paradigms that you're seeing might be shared across other tissues?
I do think that they are somewhat unique to brain because we know that the kinetics of the microRNAs are much slower in other tissue types. They tend to be pretty long-lived. They can go for 2 to 3 days in other tissue types. So I think a lot of what we're seeing is unique to neurons, and part of it, there's some evidence that it's actually the neuronal activity that can somewhat modulate their kinetics. So I do think it's probably somewhat unique to brain, but we have actually compared neurons and astrocytes, primary neurons to primary astrocytes, and the kinetics are much slower in an astrocyte compared to a neuron. So I do think I think the brain is the best thing. And
That's the best.
and I do think that some of the things we're seeing is definitely going to be specific to brain and not the other tissues.
I apologize before I ask the
Final
question.
question.
I apologize for asking this question. It's not offensive.
Okay.
But were they all told to wear dark shirts? They just happened to catch dark shirt day.
No, they planned that. They said everybody had to wear black, and I didn't, but they all decided that they were going, I said, next week is picture day, everybody show up, and then they have these group chats, and they all decided.
They all showed up that
Hmm?
way. They all showed up that way.
Oh, yeah, they all showed up, and they all, well, except Angel, but she has an excuse, so she's currently finishing her first year of medical school at UIC, but she still comes to the lab in the evenings and does work, which is pretty amazing, but yeah, they all decided that they were gonna wear black that day. So I feel like we look like we're on some kind of I don't know, TV drama show or something. It just looks, it,
Asians.
yeah, or like we're on, I don't know, like it's some sort of industry job or something. Definitely doesn't. This was just recently taken. We had a photographer come and do headshots for everybody in the department, students, postdocs, and everybody who wanted one, as well as group shots. And he put some of the labs in really weird. He did like Vogue cover shots and stuff. It was kind of fun. So.
All right.
But thank you all for your questions and your attention, and hopefully you like RNA a little bit more than you did with Toni.
Thank you.